Okay, we're in the home stretch now. This is the last section on basic limnology that I want to give to you. So a little bit more chemistry and a little bit of some biology in here. So let's get to it. We're going to talk about phosphorus, nitrogen, salinity, phytoplankton, zooplankton, very briefly. Phosphorus is not directly important to fish very much. Just to limnology, phosphorus is important because it's often the limiting nutrient, and lakes that get an input of phosphorus, say from sewage, cattle runoff, tend to be more productive because that is the limiting nutrient. Not really anything that's going to affect the fish directly. However, nitrogen can affect the fish. Now, nitrogen is also an important nutrient in lakes. Some lakes are nitrogen limited. Nitrogen chemistry is quite a bit more involved and complicated, but it is something that has some forms of nitrogen have the potential to have a negative impact on fish, and we need to understand that. Specifically, we need to worry about ammonia, which is NH3, because that is toxic to fish, whereas nitrate, NO3, is relatively safe. And the intermediate between these two, NO2, nitrite, at high levels can cause brown blood disease, but nitrite is pretty uncommon in lakes. Now, ammonia is a very common waste product of many aquatic organisms, including fish. Most all organisms have nitrogenous waste they have to get rid of. And fish and other aquatic organisms just release ammonia. But like we said, that ammonia is toxic to the fish. Well, in a typical setting, that ammonia gets oxidized into nitrate. And that nitrate is not toxic, and this is done by bacteria, and this is often called the biofilter. And so if you've ever had a fish tank, this is what the gravel's for in your fish tank. Bacteria colonize it. As the fish release ammonia, they quickly convert it into nitrate, which is safe for the fish. Of course, in the process, it consumes oxygen, so that's another source. Remember we talked about how bacteria are important for driving oxygen levels. Now, in a lake, this happens pretty easily because there's tons of bacteria relative to the amount of fish. But in captivity, when you're hauling fish, this is when your ammonia levels can start to build up and you can have potential problems with them. Now, an interesting thing about ammonia in fish is that not all forms of ammonia are toxic to fish. Well, what are you talking about? Well, you've got ammonia, NH3, and you have ammonium, NH4+. So sometimes NH3 can pick up an extra hydrogen ion. That gives it a positive charge. But in this state, in the ammonium state, that's actually not toxic to fish. Well, how come? It's just got that extra hydrogen on it. Well, that makes all the difference in the world. That makes the molecule a little bit larger, so it doesn't cross in through the gills as easily, and it's got that positive charge, which also means it does not cross the gills and get into the bloodstream. So you can have high ammonia levels, but if it's in the form of ammonium, the fish can still be fine. And the relative amount of ammonia and ammonium is driven by the pH of the water and the temperature. And so why did we talk about temperature earlier? Why did we talk about pH earlier? This is why, I mentioned that pH affects a lot of chemical reactions. This is what I'm talking about. We talked about alkalinity. Why should we care if the pH changes? Again, these things are all related. That's the thing. You got to start thinking like an ecologist and how all these things all interrelate. So if you look at this chart, this shows you the relationship between pH, temperature, un-ionized ammonia, and ionized ammonium. Okay? So, as the pH is lower, you have more ammonium, which is non-toxic. As the temperature is lower, you have more ammonium, which is not toxic. Well, this makes sense if you think about it, right? How do we get ammonium from ammonia, you add a hydrogen ion. What does pH measure? How many hydrogen ions you have. As the pH goes down, the number of hydrogen ions goes up. The more likely it is that ammonia is going to pick up a hydrogen ion. Now I'm non-toxic ammonium. As the temperature goes up, that hydrogen ion is more likely to, those reactions are going to go faster, so that ammonia is more likely to drop the hydrogen ion, then pick it up, drop it, pick it up, drop it, pick it up. So that's how temperature is going to affect it. Here's the point, is that you might measure very high levels of ammonia, but your fish look fine. And it's probably because the ammonia is in the form of ammonium. However, this can change rapidly. If the pH or the temperature changes rapidly, all that non-toxic ammonium can immediately switch to toxic ammonia, and you can get a fish kill. And that's why you need to worry about it. And so if you look at, say, a typical temperature, 20 Celsius, at a pH of 7, the relative amount, the percent of un-ionized ammonia, the percent of the ammonia that's toxic is less than 1%. Now that's still, it does not take much ammonia to kill a fish. But at 20 Celsius and a neutral pH, less than 1% of all the ammonia is in that toxic form. But if the pH just jumps up to 8, look, it triples. It goes up to at least 3%, which is plenty of ammonia to kill fish. And so this happens in hauling or when you're holding fish and you've got a lot of the non-toxic ammonia and you do a big water change, which causes the pH to jump up, and now the remaining ammonia, which 5 minutes ago was not toxic, now becomes toxic because of the pH change. So that's how you gotta think about all these things at once. All right, another thing about nitrogen is the gas form, which is N2, which is all around us in the atmosphere, dissolves pretty well in water. And so you can have nitrogen gas in water. And it's dissolved and it's fine until it's not dissolved, and then it's not fine. And so nitrogen gas coming out of solution forms little bubbles. And those little bubbles get trapped in your capillaries and in your joints, and that's the bends. And this is what happens to scuba divers. You go down deep, you're constantly breathing in, your tank has got atmospheric air in it usually, which has got a lot of nitrogen. You go down deep, you've got a lot of water pressure. At high pressure, you can shove more molecules into water, so that nitrogen gas, you get more and more dissolved. And then if you shoot up to the surface very quick, all of a sudden you release all that pressure. Now that gas can't stay dissolved. You don't have as much pressure on it. So the gas comes out of solution and goes back to being a gas and forms bubbles. And those bubbles, like I said, they form in the wrong place. It's very painful and can kill you. That's what the bends is. Fish can get the same thing. If the dissolved nitrogen comes out of solution inside the fish's body, that can be a problem. Well, what could cause that nitrogen to come out of solution? I just mentioned one thing, reduction of pressure. If the fish is down deep and all of a sudden gets brought up to the surface very rapidly, all that nitrogen can bubble out. When is that going to happen? If you catch them down deep. So if you catch a fish down deep and you pull it up, then you can get gas bubble disease. You also get their gas bladder to expand rapidly and can push their stomach out of their mouth. So that's tough on them. If the water becomes heated rapidly, remember when we talked about oxygen, that hot water does not hold as much gas as cool water. And so if water gets rapidly heated, all that gas that's dissolved in the water is going to come out of solution, is going to form bubbles. So when can this happen? This can happen if you, say, bring cold water in from the ground into a hot, warm aquaculture facility. And so that cold water's got a lot of gas in it. It gets dumped into a big tank of hot water. That cold water gets heated rapidly. All that gas has got to go somewhere. It forms bubbles. If it forms bubbles inside the fish, they're bumming. Also can happen with like a power plant lake or something like that. Water released from the bottom of a dam. Pressure again. Down deep, you've got a lot of pressure, you can dissolve more gas in the water, there's more nitrogen in the water. If you've got a dam that releases from the hypolimnion, that water is going to be cold, it might be anoxic, it might have a lot of nitrogen. As soon as it's released, now all that pressure is off because it's not underneath all this water, and so those gases can come out. And if you've got fish in the water below a dam, those gases can come out inside the fish, cause embolisms in their blood or joints or a lot of things. This can happen with oxygen too. If you have your oxygen too high, this can also happen, but it's more common, I think, with nitrogen. Okay, last chemical thing I want to talk about is salinity. And we're talking about limnology, which is sort of freshwater lakes. Why we're talking about salinity? Well, freshwater lakes, they're not distilled water. They have things dissolved in them. They're ions dissolved in there. Now, if you talk about seawater is about 35 parts per thousand. And if you look at a fish salinity, if you look at all the things that are dissolved in a fish's blood, that makes them about a third of that, about 10 parts per thousand. So that affects the osmoregulation of a fish. Well, how? How do fish osmoregulate? And how does a marine fish differ from a freshwater fish? Well, you probably talked about this before in your biology classes. Freshwater fish, their inside is about 10 parts per thousand salinity. Of course, they're in fresh water, which is about 0 parts per thousand salinity. There's not much salinity there. So they are saltier than the water in which they swim. So which way is the water going to go? Into the fish or out of the fish? It's going to go into the fish, right? The water is always going to go to where it's saltier. So freshwater fish is constantly taking on water, and they have to constantly get rid of it. So they're constantly urinating. And a lot of times if you catch a fish, you pick them up and you hold them sometimes, they pee everywhere. That's because they're constantly doing that. Marine fish. It's flip-flopped, isn't it? The marine fish is still about 10 parts per thousand on the inside, but it's swimming in water that's 35 parts per thousand. So where's the water going to go? Water wants to leave the fish and go to the saltier salt water through osmosis. So marine fish are constantly losing water, so they have to constantly drink. Okay. Salinity is also related to conductivity, dissolved ions in the water. And when we talk about electrofishing, of course, that's going to be really important. So that's another chemical property we should be concerned with. Okay, last thing I want to do is super briefly just talk about some of the living things that fish depend upon. We've talked a lot about phytoplankton. We know why they're important from a photosynthesis and an oxygen production standpoint. Base of the food web. In general, the green algae are good, the blue-greens are bad. Well, the blue-greens aren't really algae, are they? They're cyanobacteria, first off. Secondly, the cyanobacteria are not as common as the greens, but if you have a freshwater system with a lot, of blue-green algae, cyanobacteria, that's going to be a more difficult environment for fish. So those blue-green algae, you tend to find them when nitrogen is limiting. Well, why is that? Does anybody know? Again, pausing. Discuss with your friends. Well, blue-green algae can fix atmospheric nitrogen. If you recall the nitrogen cycle, the atmosphere is 78%, I think, nitrogen. There's tons of nitrogen right here that I'm breathing in with every breath. But it's in the form of N2, nitrogen gas, which is a triple bonded molecule, and I can't use a lick of it. I cannot access that nitrogen to build my molecules, right? There are only certain organisms that can take that nitrogen gas, split the ends apart and fix that nitrogen into an organic form. And it's bacteria. And whether it's bacteria living on the nodules of roots of legumes on the terrestrial world, or it's the cyanobacteria living on the surface of a lake, they can fix nitrogen into organic forms, then other things can eat them, and then that nitrogen, that's the only way living things like us, non-bacteria, can get the nitrogen we need, is because those bacteria have fixed it at some point. And you say, well, that's a good thing. I appreciate that. Well, there's other reasons why those cyanobacteria are not good for a lake. But when nitrogen is limiting, if you've got an excess of phosphorus, any organism that can capture nitrogen from the atmosphere gets a huge advantage. And that's where you're going to tend to find the blue-greens. So why don't we like them then? I guess I should answer that before I go to the next one. We don't like them for a couple of reasons. One, they're not really algae, and so they don't fit, the zooplankton, the typical zooplankton that are very important to fish. Probably don't eat them very well. They've got a very tough, slimy coat, so anything that eats them does not get a lot of energy. So they kind of short circuit the food web. They also give off a lot of toxins. And there have been reports of dogs swimming in farm ponds that have a blue-green algae, and the dog dies. And these things are making you sick, so they're not something that makes it a pleasant environment for fish and for humans. You also see filamentous algae. Some people call this moss. This is the stuff you see growing on the bottom, floating at the surface. This too is a green algae, but it's not necessarily desirable for fish. Again, it's big, it's heavy, it's tough. There's not a lot that eats it. Grass carp will eat it. And so it kind of short circuits the food web a little bit. So what we really want are those green unicellular phytoplankton. That's what drives the aquatic ecosystem. Okay, last thing we need to mention, of course, is the next trophic level up, which is the zooplankton, and then ultimately the inverts. These are the herbivores that eat those phytoplankton and then get eaten by other zooplankton, other inverts, or other fish. And all fish at some stage in their life are going to rely upon zooplankton and inverts. A lot of them, it's just during their young juvenile stage, larval stage, and then they grow up large enough to start becoming carnivorous. A lot of fish are zooplanktivorous throughout their entire life. Very few fish are herbivorous, so very few fish can eat the algae directly and get energy from them. Even some of the fish, like silver carp, that eat the algae directly might not get energy from the algae themselves. They might get energy from the bacteria that are eating. There's bacteria in their gut that eat the algae, and then they digest the bacteria. So there are very few fish herbivores, so everybody else at some point requires zooplankton inverts. And so those also become very important to the fish. Hey, what do you know? That is Limnology in a nutshell. Please understand, we went over things really quickly. We could go into a lot more detail. Everything I gave you is easily stretched into an entire semester course. What I wanted to do is talk about the important things that are going to influence fish, and this is a fish techniques class, or this might be for fish management too. And so these concepts that I've talked about in this limnology section are stuff that we're going to be calling back as we do things in the future. So let me know if you got any questions. Thanks for listening, and I'll talk to you later. See ya.